I want to welcome everybody to our first actual official horticulture seminar of this semester. Many of you were able to join us at the Genomic Selection Symposium and hear Dr. Jeffrey Endelman from the University of Wisconsin-Madison to speak about his potato breeding program. But today is actually the kickoff of our departmental seminar series. And so we're very happy to have Dr. Umesh Reddy here from West Virginia State University. He's a professor, works on a lot of different vegetable crops, works on peppers, but has many other, works on a lot of cucurbits and different species like that. He was, for those of you who don't know, Umesh actually did a postdoc here back in 1998 to 2000 with Alan Pepper, worked on cotton breeding, and he continues to collaborate with the USDA cotton group here at in College Station. Before I let Umesh get up and present his talk, I do want to mention a few things. If you notice, you came in and there's no refreshments here today. Well, there are refreshments, but what we're going to do something a little different this year. We're going to have our refreshments after seminar, because in the past, we would always invite everybody to go out and have a drink and mingle and get to know the seminar speaker, but many of the students don't have cars, and many of the students are uncomfortable, going far from campus. They don't know where the Fox and the Hound is and that sort of thing. So what we're going to do we're going to try this year is we're actually going to have our refreshments afterwards, so that you get a chance to interact with your colleagues and also with the seminar speaker in an informal atmosphere. You don't need to worry about having a car to get home and that sort of thing. And then Umesh will probably stay for as long as people have questions and want to talk to him, and then he'll be taken out to dinner by Dr. Patil tonight. But hopefully, they can stick around for 30 or 40 minutes and everybody can enjoy refreshments after the seminar. So that's the first change. The second change, which I didn't think was going to happen, is that we have asked our students to actually introduce the speakers. And so that will happen, even though I gave you a little bit of background about Umesh. We want our students to have the opportunity to get more experience with their speaking skills, being comfortable, networking with seminar speakers that come into town or those that are in different departments around campus. And so since I'm hosting Dr. Reddy while he's here, my student, Nolan Bentley, has agreed to get up and give a brief bio. So that's another change. So if you are hosting a seminar speaker, I know some of you are in the room that are hosting seminar speakers, what we would like to ask you to do is to recommend to one of your students to get up and provide the background or bio for that week's speaker. If they're not comfortable or you don't happen to have a student that can do that, I would ask you to contact Nolan, who's the president of the Hort Graduate Council, and he'd be happy to find some other student who will very readily volunteer for that activity. Okay? And then finally, one final announcement. Remember, tomorrow evening is the HGC Chili Relleno Fest in the Howdy Gardens. And I signed up, and I noticed I am the only faculty member that has yet to sign up for that. You're not on the list, though. So, and we have a lot of students who are coming, and this is a great opportunity for the faculty to interact with the students and get to know students that aren't in their program. And so I would highly suggest that everybody, if you haven't signed up, go ahead and sign up so that Holly and actually Nolan know how many people to plan for, because obviously they're going to be stuffing chili peppers and cooking and things like that. They kind of need to know how many to plan for. And then in association with that, Holly is planning the Grape Stomp Fest to raise money for the charitable campaign contribution. So bring a white t-shirt, or you can buy a t-shirt for nope, you can't buy a t-shirt anymore. Okay, bring a t-shirt if you want to dye your t-shirt. She's getting all the grapes and everything ready. So with that, I'm going to turn it over to Nolan, who's going to introduce Dr. Reddy.
Hi, I'm Nolan Bentley. So Dr. Umesh Reddy is a professor of biology at West Virginia State University. His research uses techniques techniques like genome-wide association mapping and QTL analysis to research fruit quality and shape in peppers, watermelons, and pumpkins. Dr. Reddy obtained his Master's of Science in Genetic and Plant Breeding at Tamil Nadu Agricultural University in Coimbatore, India. He then earned his PhD from the Department of Genetics at Osmania University in Hyderabad, India. He then experienced the highlight of his career as he completed a postdoc position here at Texas A&M in Dr. Alan Pepper's lab studying cotton. Then he got an assistant professor position at Alcorn State University, and then in 2004 he joined West Virginia State University, where he currently is. So during the seminar today, he will be comparing the analysis of genome-wide divergence, domestication footprints, and genome-wide association when studying fruits, traits, and peppers. So, thank you guys, and Dr. Reddy.
Thank you very much, Nolan, for a great introduction. To start with, I thank Dr. Klein and other seminar committee for inviting me here, and it is pleasure to visit College Station to see my mentor and other friends here. Thank you very much for the invitation. So, as Nolan completely read my title, it is. Yeah, can I? Oh, this is maximum it goes? Okay.
You may need to move it up just a little bit.
Oh, this is the, okay, okay. No, that's fine. Okay, so do I need to press something here for recording? You said, is it recording already? Okay, thank you. So the title is Comparative Analysis of Genome Divergence and looking for domestication footprints in these two species. One is Capsicum annuum and other is Capsicum baccatum. So the genus Capsicum is named because this genus can accumulate this molecule capsaicin, which provides pungency in the pepper fruits. So the Capsicum annuum, the most diverse species, most diverse cultivated species in the Capsicum genus, it has quite a bit of diversity. The fruit sizes wild to domesticated, and the wild itself has a high amount of capsicum, so the very hot peppers. But once it became bell peppers, in bell peppers there is no capsaicin. So probably the Capsicum genus arose by recruiting this new pathway, capsaicin, and then the breeders generated the capsicum varieties with completely no capsaicin and increased fruit size. So this is another cultivated species which cannot be crossed with annuum, with some exceptions. Very few accessions of baccatum can be crossed with annuum. More or less it is highly incompatible with annuum, and it has tremendous fruit diversity as well. So we just would like to know what is happening with these two species and what is the extent of divergence, and what is whether the domestication footprints are the same or different, and also how we can use genome-wide association studies to generate fruit-specific or other important markers. So the family is Solanaceae, and it's a pretty old crop, 9,000 years old. And Mayan cultures, Mayan civilizations, a lot of utensils that they excavated had remnants of pepper in their food. So this is one of the ancient food crops. And it is diploid and self-fertilized, having 12 chromosomes, and originated in South America and Central America. The pepper is a very good spice and a food crop and also a medicine. And you all know the crops will have the pepper spray, and then can be used as deterrent and insecticide and pesticide as well. and pretty good preservation, and will have town names, rituals, tribute. In India, if you buy a new car, you will tie a couple of peppers to the car so that the people will not have bad sight on your car. Okay? So when you buy a new car or new something, you can tie a couple of peppers there. And then it is domestication syndrome in the sense how it is different from the wild. So in the wild, the fruits are small and round, and after maturity, the fruits will become red in color, and then peduncle abscission happens at maturity, so most of the fruits would drop upon maturity, and then having high capsaicin levels. Whereas in domestication, the fruit is quite large and altered fruit shapes. You have seen the bell peppers and different kinds of serrano peppers, jalapenos, everything different shaped peppers. And then the retained peduncle at maturity. That is one of the most important domestication trait. Like grain shattering used to happen in cereal crops. And once the domestication happened, the shattering is shattering won't happen in domesticated cereals. Similarly, in peppers, retained peduncle on maturity is one of the important trait. And then variable capsaicin levels. So the genome size is comparatively, these are three different cultivated groups. So this is called annuum complex, in which annuum is there, and also Tabasco peppers, that is Capsicum frutescens and chinense, habaneros. So all these three are called annuum group. And within baccatums, baccatum variety pendulum, the pendulum is the one that has long peduncle, is one of the domesticated peppers, whereas baccatum and baccatum amblicatum are wild. Okay? So in the eximium complex, the pubescens is the cultivated pepper, and whereas the rest of them are wild. So you can see the variation in the genome size, that is 3,273 megabases to 5,655 megabases. Okay. So the peppers originated somewhere here and spread to South America, Central America, as well as North America, and domesticated in respective places. Especially the baccatum domesticated somewhere here in South America, whereas annuum domesticated in Mexico, and habaneros domesticated in Central America. So you can identify more or less the species looking at the petal color morphology. Annuum has white colored flowers, whereas baccatum has yellow spots, white flowers with yellow spots. And it's a lot of exceptions can be there. You can find slight modifications in colors, but more or less all annuums are white colors. And whereas eximium complex has purple colors. So the objectives of study, as I said, is comparing the divergence levels and looking for the domestication footprints, and to perform genome-wide association studies to identify markers or genes for horticulturally important traits. So we performed a technique called genotyping by sequencing, which is trivial these days since inception of next-gen sequencing. So the genotyping by sequencing is a very simple technique. If you have done in AFLPs previously when genotyping started, the AFLP starts with digesting genomic DNA with a restriction enzyme and ligating the adapters. And then slightly the procedure is different in AFLP, that the primers will be selected to size select the fragments. But here, digesting with the genomic DNA, and the fragments will be ligated with the enzyme-specific adapters and the barcodes. So since the unique barcodes are there, can be coupled with the adapter sequences, all the genotypes, once they are digested, adapter ligated, barcoded, can be mixed together and then sequenced together, all of them. You see in this tube different color spots are different accessions containing different barcodes. And once you do the sequencing, the reads of sequences can be aligned, and you can look for variants. They are the SNPs. That's how the large number of SNPs can be obtained using the genotyping by sequencing. So as the name indicates, it is a genotyping technique, but by just doing sequencing. So this is the total number of SNPs we got per chromosome. So once we did the GBS, all the reads could be mapped to the whole genome sequence of the pepper. And then we could identify the chromosomal locations for all these peppers. You can see the total number of SNPs per each chromosome, like 7,678 SNPs on the chromosome number 1, totally 66,960 SNPs. So these are just a total number. But once we reduce it, the minor allele frequency, that is out of 100, 5 of the variants will have this mutation. So that mutation is not sequence error that prevailed in the population. It is doing something there. So if we do the cutoff of minor allele frequency of 0.5%, or 5 out of 100 genotypes maintaining the mutation, so that we can easily believe, okay, this is definitely an allele, because at least 5 genotypes out of 100 are showing up. So that kind of stringency applied, the number of SNPs are drastically reduced, but still we have 7,000 good SNPs. Okay, and with the call rate of 90 and above. So again, the call rates will differ because this is the random sequencing. So all the accessions will not get sequenced. So the SNPs that are present at least 90 and above % of accessions are 7,331. So 10% of them roughly. And then SNPs that are in the exons of various genes are 2,521. So our study, in summary, that is one SNP is present at every 40.7 kb, that is before filtering, and then one SNP at every 78.8 kb in the coding regions, that is the exons. Whereas once you apply this minor allele frequency, that is the SNPs that prevails at least 5 individuals out of 100, then one SNP is at every 172.2 kb, and then in exons it is one SNP at every 386 kb. So we have genome-wide SNPs. So based on using all the SNPs that have minor allele frequency, that is 7,000 SNPs were used for making this neighbor joining analysis of both species, Capsicum annuum and Capsicum baccatum. So here is the total analysis, and then here is the baccatum cluster, and this is the annuum cluster. You see, several wild species that belongs to baccatum and annuum are in between. I think the wild species of baccatum and annuum, they freely cross. There are a lot of intermediate wild species. Whereas in baccatum, there are 3 different clusters. The annuum has 2 different clusters. So this annuum cluster is sweet peppers or bell peppers, and this is the robust cluster with hot peppers. So this is the principal component analysis with the SNPs that have 0.05% minor allele frequency, and there is another stringency called LD pruning. So I will explain what LD is. If the SNPs are in high LD, we don't need to pick all of them because all of them will give the pretty much same information. So we can remove the SNPs that are in the same LD block. So once all that reductions are done, with this principal component analysis suggests baccatum has 3 different clusters, and here is the annuum cluster, and several wild species are in between. And then here you see there are 3 baccatum accessions that are almost closer to the annuum. Though the baccatum and annuum are incompatible, some of these species, some of these accessions that are closer to annuum can be probably useful for making crosses. And you see some of the wild species of annuum and wild species of baccatum are clustered. So there are, even in the subclusters of baccatum, there are some wild species. This triangle shape is wild species, and those dots are the cultivated. So what it tells us that initially there was one wild ancestor and that diverged into wild species themselves diverged into wild baccatums and wild annuums, and wild annuums gave rise to the cultivated annuum peppers, and wild baccatum gave rise to the cultivated baccatum peppers. So within annuums, minor allele frequency is slightly larger, 11K SNPs. And then here you can see these blue spots are the wild species. And these are other colors, these red colors are the cultivated. And here are two wild species. They are tepin, TEPI and tepin, which is very famous wild species of annuum. Okay, that is what recently Chinese sequenced it along with a cultivated annuum species. And then there are some cultivated species that are closer to tepin. And here are some wild species that are far away from the tepin. They are called bird's eye baby. Bird's eye baby. Okay. So there are several descendants of bird's eye baby related wild peppers, and some cultivated species are around the bird's eye baby. So it tells us there may be multiple domestications in Capsicum annuum, not one single domestication. Whereas, sweet peppers are far away from the wild, because all the domesticated peppers that are close to the wild are hot peppers. So here is same principal component analysis that is grouped based on the capsaicin contents, pungency levels. And you can see here all the sweet peppers, these blue colors are sweet peppers that are grouping together. And then the hot peppers have two accessions. One is probably coming from the bird's eye baby, and the other domesticated hot peppers are coming from the tepin, two wild resources. And then we did GCMS analysis of these peppers. We don't have a lot of nutraceutical knowledge, but we did GCMS and we got about 5,000 compounds. And then we looked for the compounds that are in the capsaicin pathway. So we got the quantities of different metabolites in the capsaicin pathway for various Capsicum annuum accessions, and did principal component analysis. That also separates sweet peppers, hot peppers, and moderate peppers into different groups, just like SNP data. So capsaicin is one of the most important discriminatory trait in peppers. And similarly, we did another analysis, trivial analysis everyone does, is to understand if there is any population structure in the population that we studied. The population structure shows that there are 4 different substructures in the population. And then those 4 substructures, if we study with the context of capsaicin content, we have highly pungent peppers grouping into one cluster, and moderately pungent peppers in the medium group, and low pungent peppers in another group. And similarly, the fruit weight corroborates with the same. Capsaicin and fruit weight go hand in hand. Here, the smaller peppers are highly pungent, and the medium-sized peppers are more or less medium capsaicin, and then the low capsaicin peppers are large-sized. So this is another important analysis. This is nucleotide diversity. How nucleotide diversity can be estimated? That is taking the average pairwise differences. Like if we have 4 accessions, A, B, C, D, A versus B, and B versus C, and C versus D, D versus A. In all combinations, you can estimate average pairwise distances and then mean it. That is called pi. Whereas there is another nucleotide diversity, that is theta. Theta is more like expected nucleotide diversity. Pi is observed nucleotide diversity. So the theta is estimated based on the segregating sites, total segregating sites. So there is difference, same nucleotide diversity. One is observed and expected. So what is if the observed and expected are exactly same, what could happen? What should be the population property? Can anybody guess what should be the nature of population if theta and pi are equal? That means the population is in equilibrium, Hardy-Weinberg equilibrium, where no selection is happening, no migration is happening, and random mating is maintaining genes and genotype frequencies from one generation to the other, right? That kind of theoretical population where pi and theta will be equal. But here it is different, as pepper is one of the cultivated species. Farmers cultivate, plant breeders select, so the pi and theta would differ. So what would that information will tell us? We can estimate Thajima's D. So what is Thajima's D? That is the difference between the observed and expected diversity. Not just difference, it is normalized, little bit more presentable. So here it will tell us if Thajima's D is positive, what should we think about it? Or if the Thajima is negative, like in baccatum, what should we think the population is? Here it is all baccatum, that is the nucleotide diversity of baccatum and the expected nucleotide diversity of baccatum and Thajima's D. You can see here all different bars are different chromosomes. See, each bar is a chromosome. There are 12 different chromosomes on pepper. Here most of the chromosomes in baccatum is negative. So that would tell us that the baccatum domestication has undergone substantial bottlenecks. Probably the domestication happened in baccatum from very sizable wild species or very sizable land races. Whereas in annuum, there are different several domestication events. That's why the population is maintaining the diversity. Okay, this is the cultivated population, by the way. And all are positive except on one chromosome, and we do not know why this chromosome is undergoing completely, purifying selection comparatively to the other chromosomes. So this kind of analysis will tell us evolutionary dynamics of the population. And here, a chromosome is accumulating a lot of new and novel alleles. So, these two species are incompatible. You cannot cross them. Now you know why. Because their evolution is totally different. The evolutionary, the kind of bottlenecks the population have undergone, the domestication itself diverged these two species, which is why you cannot cross them. So here is another parameter, that is FST. FST is nothing but a fixation index. That is, if any allele is very important or favorable, and that gets fixed in the population, okay, that will get into fixation. So the FST is fixation index. So when we do the pairwise FST estimations, if the FST is very high, then we think these two populations diverged quite a bit, okay, differentiated quite a bit. Here you can see the difference between the domesticated annuum and the domesticated baccatum having 0.78 FST. So again, wild annuum, here is the wild annuum, here is the wild baccatum, still has very high FST. That tells us clearly that the wild species, the ancestral wild species, split into baccatum group, that is wild baccatums, wild annuums, before they got into domestication. Okay? And then there are other kinds. There is crown-shaped baccatum, and this FST tells us they are very close to wild baccatum, as well as crown versus cultivation. Here, 0.03. So they are pretty much close in between domesticated baccatum and wild baccatum, the crown-shaped fruits. I will show you how the crown-shaped fruits looks like. So here is chromosome-wide FST analysis. So wherever you see the FST peaks, what do you mean by having this lot of fixation index here? There are a lot of favorable genes. There are most important genes for pepper domestication, because these are pairwise FSTs. Here is baccatum cultivated and baccatum wild, and pairwise FST of cultivated annuum and wild annuum. So the peaks will tell us there are alleles that are important for domestication in those near those peaks. Then interestingly, you can see most of the peaks you can compare between these domestications. This one and this one are exactly same. And here are some unique. This peak is not there in the annuum. In the baccatum, this peak is trivially there in the annuum. So there are some unique fixation indices in both species. And then we can look for the selective sweeps. Here on chromosome number 11, we identified a huge selection sweep. In the sense, positive selection causes this kind of sweeps. So if any important allele is there for pepper domestication, that allele will not accumulate any mutations, because that is important for peppers. Because of those alleles, the pepper is called pepper. Otherwise, it would become something else. So that kind of most important alleles will undergo kind of selective sweeps, where no mutations will get accumulated comparatively to the neighboring areas. Whereas here you can see the relatively larger peaks, but this sweep is pretty much, there is very less nucleotide diversity, and then there is a clear sweep there. So we can look what kind of genes are there in that kind of sweep. And then the genes or the nucleotide polymorphisms that are present in the sweep are in high LD. Okay, the LD is, I will discuss what is LD again in next slide. The genes that get locked, almost like there will not be recombination from one LD block, in that genomic areas of the chromosome. And here is how LD decay happens within the annuum. So LD is the most important parameter for doing genome-wide association studies. So, for example, the most important alleles specific for a subpopulation will get locked. Like whatever your valuable belongings, what do you do in your house? You put them in a locker and lock it and keep it somewhere safe. So as the genomes, the genes that are important for a population substructure, say sweet peppers, bell peppers, serrano peppers. So they all have their own important alleles that make them identity. So this kind of substructure specific alleles will get locked into blocks. So no recombination happens. So when you are crossing two serranos, these blocks move from one chromosome to the other when the meiosis happens. So no variation will be created. So why these kind of blocks are very important for performing genome-wide association studies? So if any polymorphisms we are looking in these blocks, they are static there. So you can associate these polymorphisms. If you can associate these polymorphisms with any traits, those associations are robust. So the philosophy behind the genome-wide association study is, first you need to figure out what are the LD blocks, what is the extent of LD block in the genome, and accordingly you identify the polymorphisms, and you associate those polymorphisms with various traits. That is the philosophy of genome-wide association studies. If there is no LD, the markers are freely, the alleles are freely crossable throughout the genome. That means that population is kind of theoretical population, like Hardy-Weinberg equilibrium, where not much is going on. And there is no way you can do association study there. Okay? There has to be some selection, and this selection has to fix some alleles in these LD blocks. So this is another way of looking LD blocks, that is within the block, string of nucleotides are called haplotype. So when the recombination happens, when you cross two diverse peppers, for example, if you cross bell pepper with hot pepper, the hot pepper specific haplotype or bell pepper haplotype will get swapped. So the whole haplotype is intact because it is a block as a LD block. Okay. So you see here the LD pattern in the domesticated annuum. So each color is different chromosome. See, almost whole chromosome is under LD, whereas here and there some gaps. So that is the decay of the LD. So in the previous slide, we have seen 100 kb. Here, the decay happens at 100 kb. These different spots are different chromosomes, whereas if we group these SNPs into haplotypes, you can see how the LD is decaying much more earlier, that is 20 kb at the haplotype level. But within the genes, I have told you we have some exon-specific genes. If we estimate LD blocks within the genes, the LD decays quite fast, that is 2 kb. So here, you can see quite a bit of LD in the cultivated annuum types. So that means you don't have to have a whole lot of millions of SNPs for performing association studies in annuum. Like in maize, LD decays at 5 kb. I do not know how LD decays in sorghum. Right. So if we have the large LD across the genome, then we don't have to have a lot of SNPs, such as in pepper. Whereas in maize, when the LD decays at 5 kb, if you need to identify useful markers or useful genes, you need to have millions of SNPs. So that way, the cultivated annuum is very good material for performing genome-wide association studies. And there is another population genetic parameter that is called runs of homozygosity. You know, human geneticists repeatedly use this to understand deleterious alleles. In human genomes, like if we undergo inbreeding, our deleterious alleles will get exposed. The heterozygosity is purged out, and then homozygosity is exposed. So here is the homozygosity. This parameter will tell us, not like the pepper breeder is selfing a few materials. It's not like that. This kind of homozygosity is in the whole breeding history, in whole population across the world. So some part of the genome is purified from the heterozygosity. Heterozygosity is purged out, and homozygosity will accumulate to the large extent. Like the stringency we put here is 1,000 kb. Okay, so even up to 1 megabase genome size is purged out of heterozygosity in these areas. So these areas are something very important for domestication. Same as in baccatum, the LD decays about 100, and at the genic level, LD decays within 2 kb. In the baccatum also, you have quite a bit of haplotypes, in the sense not as intense as annuum, and also runs of homozygosity in baccatum. So using these parameters, that about 7 to 10,000 SNPs that have minor allele frequency of 0.05, and a GWAS population of 200 in annuums and 300 in baccatums, we screened for several seasons, one time even in greenhouse and 2 seasons of field. And then we wanted to perform GWAS for several domesticated traits. We think the pepper fruit size or fruit weight is one of the most domestication trait, most important domestication trait. So here is the GWAS results of fruit weight. What you are seeing is Manhattan plot, and each color is a different chromosome, and the dots are SNPs. So the SNPs that are above the bar are significant SNPs, significantly associated SNPs. Here is for capsaicin, dihydrocapsaicin, and then these are the fruit weight. So you can compare 2 field seasons of fruit weight data And you can see at the chromosome 1, here is a significant marker. And here is also, in the second season also that is repeatable. So most of the SNPs, but these SNPs that are seen in the one season, they are missing here. Whereas this SNP is conserved there, and that SNP is showing association in both seasons. So when we together summarize the number of SNPs that are associated in the season 1, and season 1 and season 2, about 28 SNPs in 2012 and 27 SNPs in 2011, whereas about 15 SNPs are common for both seasons. And similarly for capsaicin and dihydrocapsaicin that are grown in controlled conditions, about 14 SNPs are common. So similarly for fruit weight baccatum, we have done for 2 seasons, and about 46 SNPs are common for both seasons. About 45% of the SNPs are exactly showing up in season 1 and season 2. And then between the baccatum and annuum, we identified about 10 candidate SNPs that are common within annuum and baccatum, and showing similar kind of association, magnitude of association. That was very interesting. And then here is a typical fruit weight marker. Here is the fruit weight having 0 grams to 40 grams, 10 grams, 20 grams, 30 grams, and 40 grams. If on the chromosome number, if the SNP is capital A, then you have this blue color, in the sense relatively larger fruit size. Whereas this magenta color is for TT. In place of AA, here it is TT. So the TT gives smaller fruit size. So we could land onto the candidate genes of these SNPs, those 10 common SNPs that are for annuum and baccatum, and we could locate those SNPs in a gene called anchorin repeat containing protein, which is of 360 amino acids, and it is a synonymous SNP, changing cysteine to serine. Okay. So this anchorin repeats, they mediate protein-protein interaction. They are acyl transferases. Okay. So in the capsaicin pathway, like a vinyl alanine is synthesized in a pathway, and then a fatty acid extension chain is synthesized by another pathway. So this fatty acid extension chain and this vinyl ring will be attached with this acyl transferase. Okay. That marker is showing here for fruit weight. Anchorin repeat is a acyl transferase. So the same, interestingly, on chromosome 5, there are 2 anchorin genes next to each other. They are duplicate copies. One copy, one anchorin gene controlling the fruit weight size, and another anchorin gene controlling capsaicin content. That was interesting. Same gene, 2 duplicates, and one is controlling fruit weight, and the other is controlling, capsaicin. So in bell peppers, there is no capsaicin. The fruit size is larger, whereas hot pepper is small, capsaicin is very high. When we performed the qRT-PCR, the ankyrin protein that is specific to this one, it is expressing very low in the bell peppers, almost negligible expression. Whereas in hot peppers, it shows high expression, whereas the other one, the same gene, that is the duplicated copy, this shows very high expression in small peppers, and in the large peppers, low expression. So, and then it is heterozygote fixation. You see, here is a new term. These are gene families, duplicated gene families, one controlling one phenotypic trait and other controlling other phenotypic trait. It is pretty much same gene. So the polymorphisms that are accumulated in this gene are causing phenotypic variation. So this is a classical example of balancing selection. Okay, so that is the heterozygosity is maintained not at one locus, at 2 different locus, because these are the duplicated loci. The heterozygosity is kind of fixed there, just like homozygosity fixation. The heterozygosity also can be fixed when there are different gene families having duplicate genes. So this is another fruit weight gene and another non-synonymous mutation, changing amino acid from serine to threonine, and it regulates transcription through DNA binding activity. So we have another ABC transporter that is linked for capsaicin, and which is a non-synonymous mutation. So we performed GWAS for peduncle. You know, the peduncle length, that is one of the most important domestication trait, which prevents abscission at the time of harvesting. And we identified 2 candidate SNPs using GWAS, and they are really clearly showing the differences between the short peduncle and the long peduncle. And similarly here, another SNP variations showing short peduncle and long peduncle. So the most important gene underlying the peduncle length is this ring U-box superfamily protein, which is very important for cell cycle regulation. Okay. And it is a non-synonymous mutation as well, changing methionine to threonine. Here is another gene for peduncle length. It is F-box protein skip23. Okay, and its functions here, it has some proteasomal degradation of target proteins, things like that. So here is not only natural variation in the peppers. So even among the humans, we have the receptor that sends the capsaicin. For some, it is fun. For some, it is kind of poison. Okay.